So imagine looking at a towering redwood tree, right? Hundreds of feet tall, weighing I don't know how many tons.
Yeah, absolute giants.
Right. And then think about a human being, a completely different kind of complex machine, running marathons, composing music, solving math problems.
Two totally different biological extremes.
Exactly. But it is just like almost impossible to fathom that both of these spectacularly different entities started out as the exact same
A
thing.
single microscopic blob of jelly.
Yeah. One tiny cell. And the fact that this one cell somehow holds the architectural blueprint, the internal communication systems, and just the sheer biological willpower to construct a leaf or a hollow water pipe or a beating heart or dense bone, I mean, it completely defies our everyday logic.
It really does. It absolutely pushes the boundaries of what we'd even consider engineering.
How so?
Well, we're so used to building things from the top down, Like we draw a blueprint, we gather materials, and then some external builder puts the bricks in place.
Right, like building a house.
Exactly.
Yeah.
But biological life builds from the bottom up. The bricks themselves are the builders.
Oh, that's
Yeah,
wild.
the bricks actually contain the blueprint inside them.
And understanding how those microscopic bricks organize themselves, well, that's our mission for you today. Welcome to the deep dive.
Glad to be here.
We have a really fascinating, incredibly dense source document in front of us today titled Tissues in Action. It's basically this foundational, factual breakdown of how life constructs itself step by step in both the plant and animal kingdoms.
It's a fantastic text, but it is, well, it's pretty heavy on the academics.
Super dense. So we aren't just going to skim this document. We're going to act as your personal translators for this material.
No jargon left behind.
Exactly. We are going to read the original source text to you, quote by quote, leaving absolutely nothing out. And then immediately after we present the text, we're going to break it down, unpack the mechanisms, and figure out the how and the why behind these biological marvels.
Because the stakes here are just so much higher than passing a biology test.
Oh, for sure.
If we can truly comprehend how a cluster of identical cells suddenly decides to specialize, like how they communicate to become a rigid tree trunk versus a flexible muscle, we basically unlock the fundamental mechanics of life itself.
Right. So let's just jump straight into the source. The document frames this ultimate origin story right from the opening lines. I'll read how the author sets the stage.
You go for it.
Okay, quote. Life begins when a single cell divides itself several times to give rise to a large number of cells.
I want to pause right there because there's a monumental concept buried in that very first sentence.
Yeah.
The text points out that life begins when a single cell divides itself several times. Notice it doesn't say the body just gathers a bunch of different cells from the environment and glues them together.
Oh, right. Like you don't eat an apple and use the apple's cells to build your arm.
Exactly. Every single cell in your body right now is a direct descendant of that original one cell.
It's like having a single Lego block that just starts cloning itself.
That is a great analogy.
But I'll keep reading because here is where the engineering marvel really comes into play. The text continues, quote, these cells gradually form the skin protection, muscles movement, bones support, nerves control and coordination, and all other organs.
Right. So if that Lego block just kept cloning itself infinitely without changing, you wouldn't get a human or tree.
You'd just get a giant formless mountain of identical red Lego blocks.
A blob.
A massive blob. But that isn't what happens. As the text explicitly states, these cells gradually form skin for protection, muscles for movement. The blocks somehow look around, communicate, and assign themselves specialized careers.
And that transition from a uniform cluster of dividing cells into specialized functional groups, I mean, that's the entire basis of what a tissue is.
Precisely. And let's hear what the text says about why we should care.
Okay, the next part says, quote, this process is so intricate that it is considered one of nature's greatest engineering marvels. Researchers have been trying to understand, replicate, and modify this process for human welfare.
Human welfare. That is the explicit motivation. Understand, replicate, and modify. Because if we can figure out the exact chemical whisper that tells a blank cell to become a bone cell, the implications are just staggering.
Staggering is the right word. You shatter your femur. We don't put a metal rod in it. We just instruct your cells to build new bone.
Or your heart valve is failing. We just grow a new one.
Right. The text is telling us that understanding tissues isn't just basic anatomy. It is the ultimate key to regenerative medicine.
Okay, finishing this first section's text, quote, to do so, it is essential to understand the natural biological processes that govern growth and development in plants and animals. What makes cells group together to form tissues? Why do some tissues grow throughout life while others do not?
And to answer those questions, the author recognizes we need to build a mental model. Like, how does life actually scale up from the microscopic to the macroscopic?
Which brings us to the next section. It lays out this architectural ladder of life. Let me read the text's exact breakdown.
Let's hear the hierarchy.
Quote, in Chapter 2, Cell, the building block of life, you have learned that the cell is the basic unit of life. Many cells come together to form a multicellular organism. In all multicellular organisms, there is a hierarchy of organization.
Hierarchy of organization, that's the key
I'll
phrase.
read the steps it gives. Quote, cells of similar type performing similar function group together to form a tissue. More than one type of tissues form organs. Different organs form organ systems, and organ systems form an organism.
Okay, let's extract that core mechanism. The text gives us a very clear five-step ladder.
Step 1 is the cell, the basic unit.
Right.
Step 2 is the tissue, which the text defines as cells of a similar type performing a similar function.
Step 3 is the organ. So more than one type of tissue coming together, like your heart is made of muscle tissue, but also nervous tissue and connective tissue.
Step 4 is the organ system, the heart working with the blood vessels.
And step 5 is the organism. You, me, the redwood tree.
But what I think is crucial to understand is why this hierarchy even exists. And the text actually uses a brilliant contrast to explain it. Let me read the next lines.
Go ahead.
Quote, in unicellular organisms, such as amoeba, a single cell performs all functions of life. In multicellular organisms like plants and animals, different groups of cells perform different functions.
So it brings up the amoeba. An amoeba is a unicellular organism, just one single cell.
Right.
And as the text notes, that one cell has to perform all functions of life.
I always look at the amoeba as like the ultimate biological solopreneur.
A solopreneur.
Yeah. It's a one-cell startup. That single cell has to hunt for food, it has to digest the food, it has to expel waste, move around, defend itself against predators, and reproduce. All by itself.
And being a solopreneur is incredibly taxing. When you have to do absolutely everything yourself, you can never truly excel at any one specific task.
Right, you're a jack of all trades, master of none.
Furthermore, there's a hard physical limit to how big you can get. An amoeba can never grow to the size of an elephant because a single cell physically cannot manage the logistics of a body that large.
So the evolutionary solution is what the text calls the division of labor. Let me read that exact part.
Please do.
Quote, a tissue is a group of cells similar in structure that work together to perform a specific function. The formation of different types of tissues leads to the division of labor, which increases the efficiency of the body and enables it to carry out complex life processes.
Division of labor.
Yeah, when you have a multicellular organism with trillions of cells, they don't all have to hunt, digest, and move. They can specialize.
It's exactly like a modern city.
Right. You don't have every single citizen farming their own wheat, building their own house, and generating their own electricity.
No, you have farmers, construction workers, and power plant operators.
Yeah.
That specialization, which the text points out, is the very definition of a tissue, is what increases efficiency.
The examples the text provides highlight this beautifully. Let's finish this paragraph. Quote, for example, in animals, muscle tissue enables movement and nervous tissue carries messages to different parts of the body. In plants, conducting tissues such as xylem transports water and minerals, while phloem transports food.
See, muscle tissue doesn't have to worry about digesting food at all. Its only job, its entire structural focus, is contracting to enable movement.
Nervous tissue doesn't worry about moving. Its architecture is entirely devoted to just carrying messages.
The cells have optimized their physical structure for one highly specific task.
Form follows function. And the text teases the plant examples too, xylem for water and phloem for food. We are going to spend a lot of time on those biological pipelines shortly.
We definitely will.
But first, the source document gives us a moment to consolidate our thinking. It provides a section called Think It Over with 3 specific guiding questions. Let me read the exact text.
Let's hear them.
Quote, think it over. How is the study of cells and tissues significant for understanding the life processes and human welfare? Next question. How are tissues in plants and animals different, and why? And the third question. How is the division of labor at various levels of organization in multicellular organisms correlated with their structure and function?
These aren't just review questions at the end of a chapter. They are basically the architectural framework for everything we're going to explore today.
Yeah, they really set the roadmap. We've just touched on the first and third questions, like We know tissues are key to human welfare because they are the blueprints of regeneration.
And we know the division of labor correlates with structure because a cell's physical shape is dictated by its specialized job.
Which leaves that second question hanging right in the middle. How are tissues in plants and animals different, and why?
And that brings us to section 3.1 of our document.
Exactly. We're going to look at the great divide between the two major kingdoms of complex life. Listen to how the author boils down the fundamental differences between flora and fauna. I'll read the first part.
Go for it. Quote, 3.1. Why are plant and animal tissues different? In Chapter 2, Cell, the building block of life, you compared plant and animal cells and studied the major differences between them. Most plants are fixed in one place and do not move from place to place like animals. They need support to stay firm and upright.
Okay, let's stop there. The text distills the immense complexity of biology into two really pragmatic, real-world constraints: lifestyle and diet.
Lifestyle and diet.
Let's tackle the lifestyle aspect first. The text points out a seemingly obvious fact. Most plants are fixed in one place and do not move.
Right.
But the structural implications of that are just staggering. If you are a plant, you are rooted to a specific coordinate on the earth.
You cannot run away from a storm.
No, you can't. You cannot seek shelter from the wind. You have to endure gravity constantly pulling down on your branches 24 hours a day without ever sitting down to rest.
So the primary engineering requirement for a plant is structural integrity.
Exactly. They need support to stay firm and upright. And let's see how the text explains how they achieve this. The next line says, quote, "Plant cells have a cell wall that provides rigidity and strength."
A cell wall. This isn't just a soft membrane. It is a literal physical wall built around the perimeter of the cell.
It's as if every single cell in a plant is encased in a microscopic cardboard box.
Yes.
And when you stack trillions of those rigid boxes on top of each other, you get a structure that can withstand hurricane force winds.
But then the text pivots to animals. Let's read what it says about them.
Quote, "In general, animals can move, although some, such as sponges, are immobile. Without a rigid cell wall, animal cells can change shape easily. This cellular flexibility eventually helps make their bodies suitable for locomotion."
Movement requires something entirely different from rigidity. Movement requires a dynamic shifting of mass.
Right, if animal cells were encased in rigid cardboard boxes, our muscles could never contract.
Our joints could never bend. We'd just be completely immobilized statues. So, the text notes, animal cells are completely missing that rigid cell wall.
And this lack of a wall is what the text calls cellular flexibility. It allows an animal cell to squish, stretch, and change shape.
And that microscopic flexibility is what scales up to allow a cheetah to sprint or a human hand to play the piano. It makes the body suitable for locomotion.
Though I do want to highlight a wonderful little caveat the author included in parentheses there.
The sponges.
Yeah. Quote, "Although some, such as sponges, are immobile." It's a great reminder that biology just loves to blur its own lines.
It really does. It feels like a paradox, doesn't it?
Totally. If a sponge just sits there anchored to a rock on the ocean floor, exactly like a plant, why on earth do we classify it as an animal?
Well, it comes down to the deep cellular architecture. And the second major difference, the text highlights nutrition. Sponges, despite being stationary, do not have rigid cell walls. And more importantly, they cannot make their own food from sunlight.
They have to eat things.
Right. They have to capture and consume external food particles drifting in the water.
Which perfectly segues into that second massive divide in the text. Let me read the rest of the section. Quote, "Another major difference between plants and animals is their mode of nutrition. Animals have tissues that help them digest food obtained from different food sources, while plants have tissues that help them utilize solar energy for synthesizing the food components through photosynthesis."
Animals are basically biological combustion engines.
We really are.
We have to constantly roam around, find external fuel sources, consume them, and break them down. That requires an incredibly complex suite of digestive tissues, acid-secreting organs, and absorptive linings.
While plants, as the text says, utilize solar energy, they have evolved the ultimate energy independence. They just sit in the sun, open their microscopic pores, and literally build their own food out of light, water, and thin air.
It's incredible. And because they acquire their energy in such fundamentally opposed ways, their internal plumbing has to be completely different.
The text emphasizes this. Quote, "Plants and animals have distinct tissues for transporting food and water to different parts of the body."
Right, an animal needs a muscular heart to pump nutrient-rich blood to hungry, moving muscles.
But a plant needs a passive, gravity-defying system to move water from the deep earth up to the sunlit leaves.
And as the final sentence of that section points out, this also drastically alters how they grow. Let's hear it.
Quote, "The growth patterns in plants and animals also vary because the tissues responsible for growth differ in structure and function. In this chapter, we will learn how the structures of plant and animal tissues relate to their specific functions."
An animal grows uniformly to a predetermined shape and size, and then largely stops.
Yeah, I stopped growing in high school.
Exactly. But a plant's growth pattern is entirely different because it never really stops exploring its environment for more light and water.
Which is the perfect entry point into the next major concept. If plants are permanently fixed in place, encased in rigid microscopic boxes, how do they actually grow?
Right, how does a tiny seedling push through the heavy soil and eventually become a massive tree?
The source text dedicates section 3.2 entirely to the specialized growth engines of plants. I'll read the introduction to the section.
Go for it.
Quote, 3.2 Tissues for growth in plants. You must have observed that a small seedling grows into a tall tree, roots grow deep into the soil, stems become thicker with time, and grass grows again after being eaten by grazing animals. Which tissues are responsible for these changes?
The author is establishing three very distinct physical challenges the plant has to overcome here.
Let me read the bullet points the text gives for those challenges. Quote, plants grow in different ways. Increase in length, height of stem, and depth of roots. Increase in girth, thickness of stem, and regrowth after cutting the branches or grazing by animals.
So it needs to reach higher and dig deeper. That's length. It needs to reinforce its structural column to support that new weight. That's girth or thickness.
And it needs a contingency plan for physical trauma, like an animal taking a bite out of it, which requires regrowth.
And the text provides a single, unified answer to all three challenges. Let's hear the next sentence.
Quote, this growth require actively dividing cells that together form a tissue called a meristematic tissue. Let us explore different kinds of meristematic tissues.
Meristematic tissue.
Meristematic.
If you take nothing else away from plant biology today, remember that word. The text defines it simply as a tissue formed by actively dividing cells.
I think of meristematic tissues as the dedicated construction crews of the plant world.
Yes.
Like if you look at a mature leaf, those cells are largely done growing. They are just doing their job, photosynthesizing.
They're just living their lives.
Right. But hidden in specific pockets around the plant are these meristematic zones. These are clusters of cells whose entire existence is devoted to relentless, continuous division. They are the factories churning out new microscopic bricks.
And the plant strategically places these construction crews exactly where growth needs to happen. The text breaks down these specific locations, starting with how plants increase in length.
And to explain this, the author introduces an experiment. It's an incredibly elegant way to visualize something invisible. I am going to read the exact setup and execution of Activity 3.1.
Oh, the onion experiment.
Yes, this is great.
Quote, 3.2.1, Apical meristem. How do plants grow in length? Let us study the growth of roots in an onion bulb. Activity 3.1. Let us design experiments. 1. Take 2 glass jars or Cuplin jars and fill them with water. 2. Now, take 2 onion bulbs and place 1 in each jar, as shown in figure 3.1.
Okay, simple enough so far.
Quote, 3. Observe the growth of roots in both bulbs for a few days. 4. Measure the length of roots on days 1, 2, and 3.
Just establishing a baseline.
And here's the twist. Quote, 5. On day 3, cut the root tips of the onion bulb in jar B by about 1 centimeters. After this, observe the growth of roots in both the jars, measure their lengths for 4 more days, day 4 onwards, and record your observations in table 3.1.
This is the scientific method in its purest form.
Yeah, you have a control group, jar A, and an experimental group, jar B. You start with 2 identical onions sitting on jars of water. Over the first 3 days, they both behave exactly the same. They wake up, they sense the water, and they start shooting little white roots downward.
But then, the crucial intervention. On day 3, you take scissors and snip just 1 centimeter off the very bottom of the roots in jar B, but you leave the roots in jar A completely intact.
Now, if you didn't understand plant anatomy, you might assume that a root grows the way a balloon inflates.
Like expanding uniformly all over.
Exactly. And if that were true, cutting the tip off wouldn't stop the rest of the root from continuing to expand and push downward.
But the text presents the conclusions we are meant to draw from the data, and it completely shatters that assumption. Let me read the text conclusion.
Let's hear it.
Quote, what trend do you observe in the data you recorded in Table 3.1? Are your observations similar to those presented in the graphical representation, figure 3.2? What do you infer? Roots in jar A continue to grow in length. Roots in jar B stop growing after the tips are cut.
They just freeze. Even though 90% of the root structure is still intact and submerged in water, the forward momentum completely dies.
Meanwhile, the untouched roots in jar A just keep plunging deeper into the water, and the text spells out the inference clearly. Quote, "This shows that roots grow only from their tips. The tips consist of cells which divide continuously."
This shows that roots grow only from their tips. The construction crew isn't spread out along the whole root. The cells that divide continuously are exclusively clustered at the very extreme boundary of the tissue.
The text continues, quote, "You may recall observing mitosis in onion root tips in Chapter 2. This observation confirms that root tips contain actively dividing cells. Similarly, shoot tips also contain actively dividing cells that help the shoots to grow in length. Thus, we conclude that plants have growth zones at the tips of their roots and shoots, called the apical meristems, which help the plants grow in length."
Apical meristems, apex meaning the tip or the peak. And the text notes they aren't just on the roots pushing down, they're also on the shoot tips pushing up into the sky.
Think about the physics of this for a moment. The apical meristem at the tip of a root is constantly generating new cells behind it.
Right.
As those new cells expand, they literally act as a hydraulic ram, physically shoving the hardened tip further down into the dense, abrasive soil.
It is a microscopic battering ram powered by cellular division.
It's brilliant engineering. But before we move on to the next type of growth, the source text actually pauses to introduce a brief cultural and historical context. Listen to the sidebar.
Oh, the Meet a Scientist section.
Yeah, quote, "Meet a scientist." B.G.L. Swami was a renowned Indian botanist known for his contributions to the area of plant morphology and anatomy. His book, Hasuru Hanu, is a popular book in Kannada language. It is a blend of science, satire, and culture. The book describes the botanical excursion in the forests of Western Ghats and is a treasure of plant descriptions, utilization, conservation practices, botanical folklores, and myths. The book won the Kendra Sahitya Akademi Award in 1978.
I really appreciate the author including this because it reminds us that the study of plant morphology and anatomy, the very tissues we are dissecting right now, it doesn't exist in a sterile vacuum.
Right. Dr. Swami won a major literary award because he recognized that botany is deeply intertwined with human culture, folklore, and even satire.
It grounds the dense science in humanity.
It does. But let's get back to the biological engineering. We know how the plant pushes up and down using the apical meristem. But a 100-foot tree balancing on a stem, the width of a pencil would instantly snap in half.
It definitely would. It has to get wider.
The text addresses this in the next section. Quote, 3.2.2 Lateral meristem. How do plants grow in girth? Look at your surroundings. You must have observed that the stems of dicot plants not only grow in length, but also increase in diameter or girth over time. What causes this increase in girth? One possible explanation may be the activity of meristematic tissues.
So we transition from vertical growth to horizontal expansion. The text uses the term girth, which simply refers to the diameter or thickness of the stem.
Let's read how it works. Quote, "If you have visited a timber yard or observed the cut trunk of a tree, you may have noticed several ring-like patterns on the cut surface of the wood. These are annual growth rings. Some annual rings are wide and some are narrow, reflecting the favorable or unfavorable growth conditions during a particular year. By counting these annual rings, scientists can estimate the age of a tree and also understand the climatic conditions under which it grew."
Okay, so it identifies the rings, but what actually makes them?
The text explains, quote, "The increase in girth occurs due to the presence of actively dividing cells arranged in a ring in the stem. These cells divide and produce new cells inside and outside in a concentric manner, leading to an increase in diameter of stem. This meristematic tissue is called the lateral meristem."
The lateral meristem. So if the apical meristem is a tiny cluster of cells at the very tip of the branch, the lateral meristem is an entirely different architectural shape.
It's a ring.
Yeah, the text describes it as being arranged in a ring in the stem. Imagine a microscopic cylinder of dividing cells running vertically just under the bark, encompassing the entire trunk.
And the mechanism of its division is crucial, right? The text states it produces new cells inside and outside in a concentric manner.
Exactly. Every time this cylinder of lateral meristem divides, it pushes a layer of new wood toward the inside of the tree and a layer of new bark-producing cells toward the outside. It is literally expanding its own waistline year after year.
And this creates a prominent physical record, which the text highlights beautifully. Those annual growth rings you see on a stump are the direct result of the lateral meristem's activity. But it isn't just a clock, it's a weather station.
It is a weather station. The text points out that some annual rings are wide and some are narrow, reflecting the favorable or unfavorable growth conditions.
So, like in a year with abundant spring rain and summer sun, the meristematic cells divide rapidly. They gorge on nutrients, creating a wide, thick ring of new wood.
But if there's a severe drought or a massive volcanic eruption blocks the sun, the tree goes into survival mode. The lateral meristem barely divides at all, conserving energy. That year's growth ring will be paper thin.
So, when the text says scientists can understand the climatic conditions under which it grew, it means we can look at a 500-year-old oak tree and pinpoint exactly which years experienced severe droughts or harsh winters centuries before human weather records even existed.
It is a phenomenal intersection of biology and climate history. So we have apical for vertical length and lateral for horizontal girth.
But the text identifies one more, highly specific growth tissue designed to solve a very particular threat.
The threat of being eaten.
Right. Listen to how the author describes this third mechanism in section 3.2.3. Quote, 3.2.3 Intercalary meristem. How do plants grow after being cut? We have learned that meristematic tissues are present at the tips of roots and shoots. What do you think happens to the growth of the plant if the tip of a young stem is cut? The stem stops growing in length, but new branches arise from the nodes of stem.
The author poses a fantastic logical trap here. We just proved with the onion experiment that if you cut off the apical meristem at the tip, the growth completely stops.
So if a cow bites the top off a blade of grass, or you run over your lawn with a mower, chopping off millions of apical meristems, why doesn't the entire lawn instantly die and stop growing forever?
It's the ultimate biological paradox for anyone doing yard work.
The answer is in the next lines. Quote, the intercalary meristem is located at the base of internode or just above the node. The node is point on plant stem where branches or leaves arise. The part of stem between the two nodes is called internode. You may also have observed that when the hedge around a garden is cut, after some time more branches appear again, giving the hedge a bushy appearance. Grass also appear after some time being mowed and/or grazed by animals. This happens because of the presence of intercalary meristem at the nodes of its stem. These meristematic tissues are called intercalary meristems.
The intercalary meristem, it is a hidden backup system.
I love how it works.
To understand it, we have to look at the anatomy the text defines. It talks about nodes and internodes. If you picture a stalk of bamboo, the nodes are those solid, knobby joints where a leaf or a new branch might sprout.
And the internode is the long, smooth, hollow section between two joints.
Right. And the text reveals that the intercalary meristem is located at the base of internode, or just above the node.
It's tucked safely down near the joints, close to the ground, completely hidden from the lawnmower blades or the grazing teeth of an animal.
When the vulnerable apical tip gets severed, the plant doesn't surrender. It signals these hidden intercalary pockets to wake up. They start dividing rapidly, pushing new growth up from the base rather than pulling it from the tip.
It's a phenomenal evolutionary adaptation, specifically designed for environments where the tops of plants are constantly being destroyed.
So we have our three construction crews: apical, lateral, and intercalary. But what actually makes these meristematic cells so uniquely capable of constant division?
The text provides a powerful summary of their cellular biology and then drops a massive theoretical question right in the middle of it. Listen closely.
Okay.
Quote, thus, plants have three types of meristematic tissues: apical, lateral, and intercalary. The apical meristem, located at the root and shoot tips, increases its length. The lateral meristem, located along the circumference of stems, increases girth. The intercalary meristem, located at the base of certain plants, such as grasses, helps them regenerate after cutting. Together, these meristems account for growth in length, girth, and branching in plants.
That's a perfect recap.
And here's the biology part. Quote, the cells of the meristematic tissues are small, have thin cell walls, a large and prominent nucleus, and dense cytoplasm with many organelles. Vacuoles are generally absent, and the cells are tightly packed with little or no intercellular space. These characteristics of meristematic tissue allow them continuous and rapid cell division.
There is so much rich biology packed into those sentences. Let's start with the physical description of the meristematic cell. The text says they are small, have thin cell walls, a large prominent nucleus, and dense cytoplasm.
Every single one of those traits serves the ultimate goal: rapid division. A thin cell wall is naturally easier to split in half than a thick, armored one.
A large nucleus is necessary because the nucleus holds the DNA blueprint, and that blueprint is constantly being accessed and copied.
And the dense cytoplasm is packed with organelles to generate the massive amount of energy required to physically tear a cell in two.
But then the text throws a curveball. It notes that vacuoles are generally absent and immediately challenges the reader.
Yeah, let me read the question it asks. Quote, why do you think that the cell of meristematic tissues lack vacuoles?
This is a great moment to push back on what we normally assume about plants. If you look at a standard textbook diagram of a plant cell, the vacuole is usually the most prominent feature.
It's huge. It's a massive water-filled balloon inside the cell that stores nutrients, waste, and provides internal pressure to keep the cell rigid. Often, a vacuole takes up 80 or 90% of the entire interior volume of a plant cell.
Right, so why on earth would the most active, important growth cells in the plant be missing this crucial organelle?
Well, because when your only job is rapid, continuous division, a giant storage tank is a massive liability.
Imagine trying to pack up your apartment and move to a new city every 20 minutes.
That sounds like a nightmare.
Now imagine trying to do that while dragging an enormous sloshing waterbed with you.
It would slow you down completely. You wouldn't be able to move fast.
Precisely. Meristematic cells are the ultimate minimalists. They do not have time to store vast amounts of food or manage large quantities of waste. They burn energy as fast as they get it to fuel the division process.
Having a massive vacuole would physically get in the way of the complex machinery required to split the cell's chromosomes. They're built purely for speed and repetition, so they ditch the heavy luggage.
But the text also makes it clear they don't stay in this minimalist state forever. It introduces this massive biological threshold called differentiation.
Let me read that paragraph. Quote, due to continuous cell division, meristematic tissue adds new cells to the plant body. Some of the newly formed cells remain meristematic, while others lose the ability to divide. The cells that lose the ability to divide undergo changes in structure and function and become permanent tissues. These cells become specialized to perform specific functions, such as support, transport, or storage. This process, by which meristematic tissue becomes specialized to perform specific functions, is called differentiation. Meristematic tissue becomes permanent by the process of differentiation.
Differentiation is basically the biological equivalent of graduation day.
As the apical meristem pushes higher, or the lateral meristem pushes outward, it leaves a trail of newly created cells behind it in the wake of its continuous division. And the text explicitly states that some of these cells lose the ability to divide. They hang up their hard hats.
They are no longer construction workers. And once they stop dividing, the text says they undergo changes in structure and function and become permanent tissues. This is where the magic of the blueprint happens.
The cell reads a specific part of its DNA and begins to physically transform. If it's going to be a water pipe, it might elongate and reinforce its walls. If it's going to be a storage cell, it might finally grow that massive vacuole it was previously lacking.
The unspecialized, dividing meristematic cell has differentiated into a specialized permanent tool for the plant.
Which perfectly sets up our next massive segment. Once these cells graduate and take on permanent roles, what exactly do they do? The author takes us deep into the architecture of the mature plant. Let's read the introduction to section 3.2.4.
Let's do it.
Quote, 3.2.4 Permanent Tissues. Examine the transverse section of the root and/or stem and/or the vertical section VS of the leaf of any herbaceous plant under a microscope. The internal structure of a TS of a sunflower stem as seen under a microscope is shown in figure 3.7. What do you observe? Are all the cells similar in shape and size? How many different types of tissues can you identify? What differences do you notice among them? What might be the reason for the presence of different types of cells and tissues?
A lot of questions to get the student observing.
And here's the answer it gives. Quote, you can see that different groups of cells are present and each tissue is specialized to perform a specific function. These are permanent tissues. Permanent tissues can be simple, composed of only one type of cell, or complex, composed of more than one types of cells.
So we've officially left the chaotic construction site and entered the highly organized functioning machinery of the plant. And the text provides a very clean structural taxonomy right off the bat.
It divides permanent tissues into two broad categories: simple, which are composed of only one type of cell, and complex, which are composed of multiple different types of cells working together as a team.
And it begins by analyzing a simple, protective tissue, the epidermis. Let's hear what it says.
Quote, Observe the types of tissues given in Figure 3.7 based on pointers provided as follows. I. Protective tissue epidermis. What protects plants from mechanical injury, water loss, harmful microorganisms, and extreme environmental conditions? The epidermis forms the outermost layer of the plant body. It consists of a tightly packed single layer of flat and rectangular cells.
Let's pause there. The text defines this as the outermost layer of the plant body. It is the plant's skin.
And notice how the text describes the architecture of this skin. It is tightly packed and made of a single layer of flat and rectangular cells.
If you are building a defensive wall against the outside world, you don't want round cells with gaps between them. You want flat, rectangular bricks mortared tightly together so no microscopic invaders can slip through.
Exactly. But a wall of living cells is still vulnerable to drying out. So the text explains a brilliant chemical adaptation. Quote, it protects all parts of the plants. These cells are covered with a waxy layer of cutin called cuticle. In some plants living in very dry habitat, the epidermis may be covered by a thick layer of cuticle to reduce the water loss in the process of transpiration by stomata. The cuticle also provides protection against mechanical injury and invasion by parasites.
A waxy layer of cutin. It is essentially a built-in raincoat. And the text points out that environmental pressure dictates the thickness of this coat.
Like a cactus living in a scorching desert habitat will secrete an incredibly thick layer of cuticle.
The goal there isn't to keep water out. It's to desperately trap the plant's internal moisture and prevent the brutal sun from evaporating it.
But here's the profound challenge of biological design. A plant cannot be perfectly sealed inside a waxy, waterproof tomb. It is a living thing. It needs to interact with the environment, it needs to pull in water from the earth, and it needs to breathe in carbon dioxide from the air.
And the text explains how the epidermis breaks its own defensive rules to accomplish this. Let's hear the rest of the section.
Quote, in many plants, hair-like projections arise from epidermal cells. In roots, these projections are called root hair, which increase the surface area for absorption of water and minerals from the soil.
So first, down in the soil, the epidermal cells sprout hair-like projections called root hairs. Why? The text says it's to increase the surface area for absorption.
If the root was just a smooth cylinder, only the cells directly touching the soil could absorb water. By sprouting millions of tiny hairs outward, the plant exponentially increases its contact with the damp earth, acting like an incredibly efficient microscopic sponge.
But the most crucial modification happens up in the leaves.
I'll read that part. Quote, in leaves, the epidermis contains pores called stomata, which, apart from gaseous exchange, helps in transpiration, i.e. evaporation of water vapors through stomata. Thus, transpiration helps in water transportation by creating a transpiration pole in xylem. Transpiration also helps in elimination of wastes from the plant body.
Pores, called stomata. These are essentially microscopic mouths that can open and close. They allow the crucial gaseous exchange breathing in carbon dioxide for photosynthesis and exhaling oxygen.
But the text introduces a second monumental function of the stomata transpiration. It defines this as the evaporation of water vapors through stomata. It's essentially plant sweating.
And this leads to one of the most mind-bending sentences in the entire document. Thus, transpiration helps in water transportation by creating a transpiration pole in xylem.
We really have to unpack the physics of this transpiration pole because it borders on the miraculous.
It really does. Consider a giant redwood tree, 300 feet tall. Deep underground, those root hairs we just talked about absorb water. That water needs to get all the way up to the leaves at the very top of the canopy to be used in photosynthesis.
How does it get there? An animal would use a muscular heart to pump the fluid under high pressure. But a tree has no moving parts. It has no pump. How does it defeat 300 feet of gravity?
The answer lies entirely in the microscopic evaporation happening at the stomata. When the sun beats down on a leaf, a water molecule evaporates out of the stomata and turns into vapor. Because water molecules have a property called cohesion, they are highly attracted to one another via hydrogen bonds. That evaporating molecule pulls slightly on the liquid water molecule directly beneath it.
It's like a microscopic chain link.
Exactly.
Yeah.
As the top molecule evaporates into the air, the microscopic vacuum it leaves behind yanks the second molecule up to take its place, which yanks the third, which yanks the fourth, all the way down a continuous, unbroken microscopic column of water that reaches 300 feet deep into the root system.
The tree is literally using the evaporating heat of the sun to suck water out of the earth. The physical tension inside those tiny water pipes must be immense. If you or I tried to suck water up a straw 300 feet tall, the vacuum pressure would collapse the straw instantly.
which perfectly explains why the plant requires the next set of tissues the author introduces, the internal structural supports. Let's read the section detailing the tissues designed to keep the plant from collapsing under its own physical and vacuum pressures.
Okay, here we go. Quote, 2. Supporting tissue. Simple permanent tissues. What keeps a plant upright? Why does a fresh twig bend but a dry twig break? Why are seed coats hard and how do aquatic plants float? These functions are carried out by supporting tissues. There are 3 types of supporting tissues, or simple permanent tissues: parenchyma, collenchyma, and sclerenchyma. Each differs in structure and performs supporting functions.
This section breaks down the 3 foundational simple permanent tissues: parenchyma, collenchyma, and sclerenchyma. They sound like complex jargon, but the text makes their distinct physical properties very clear.
They represent a spectrum of structural integrity, moving from softest to hardest. Let's start with parenchyma.
Let's read it.
Quote, A parenchyma. Parenchyma consists of living cells with thin walls. These cells are loosely packed with intercellular spaces. Parenchyma mainly stores food, but also performs photosynthesis in the green parts of the plants. In aquatic plants, specialized parenchyma forms air spaces, which help them float.
The text describes them as living cells with thin walls that are loosely packed with intercellular spaces.
I envision parenchyma as the biological packing peanuts of the plant. They just fill in the empty voids. Because they are thin-walled and loosely packed, they are soft. The text notes their primary job is to store food, likely holding those massive vacuoles we talked about earlier.
And in the green parts of the plant, they contain chlorophyll to perform photosynthesis.
But the text highlights a fascinating structural adaptation for parenchyma in aquatic plants.
Yeah, water lilies, for example, need their leaves to stay on the surface of the pond to get sunlight. So the parenchyma tissue in their leaves develops massive air spaces. It literally turns the tissue into biological bubble wrap, creating natural buoyancy so the heavy leaf doesn't sink.
So parenchyma is soft storage and packing. Next on the spectrum is collenchyma. Quote, B collenchyma. Collenchyma consists of living cells with unevenly thickened corners due to pectin, a chemical that gives flexibility like rubber deposition, collenchyma. The text says these are living cells with unevenly thickened corners due to pectin. And it specifically defines pectin as a chemical that gives flexibility like rubber.
This is the plant's equivalent of cartilage. If a young green stem is hit by a strong gust of wind, you do not want it to be perfectly rigid because it will snap in half. You want it to bend, bow, and then spring back to its original shape.
The pectin provides that elasticity. Anyone who makes fruit jams knows about pectin. It's the natural chemical you extract from fruit to make the jam set into a flexible, jiggly consistency. The plant deposits this rubbery substance into the corners of the collenchyma cell walls, providing dynamic, flexible support to young, growing stems.
But eventually, a plant, especially a tree, grows so massive that flexible cartilage isn't enough to hold up tons of wooden branches. It needs concrete. And that is the third tissue, sclerenchyma. Let me read it.
Please.
Quote, C. sclerenchyma. Sclerenchyma cells have thick walls due to deposition of lignin, making them hard and strong, forms the woody structure. Most of these cells are dead. This tissue is found in stems, leaf veins, and hard coverings of seeds and nuts, such as coconut husk and walnut shell.
Sclerenchyma. The text describes it as having thick walls due to deposition of lignin, making them hard and strong, and then it drops a massive biological twist. Most of these cells are dead.
This is a profound evolutionary strategy. How can a tissue be most useful to the organism after it dies?
It's the ultimate sacrifice for structural integrity. A living cell requires maintenance. It requires water, nutrients, and oxygen. But if a cell's only job is to be a brick in a wall, its living interior is actually a weak point.
So the sclerenchyma cell spends its entire life pumping out this incredibly tough, rigid chemical called lignin, which is the main component of wood. It builds its own internal prison walls thicker and thicker with lignin until the living cell inside is completely choked off and dies. What is left behind is a microscopic, incredibly tough, empty casing, a biological brick. The text points out this is exactly what forms the hard, woody structure of stems, the tough, rigid veins in a leaf, and the practically unbreakable armor of a coconut husk or a walnut shell.
It is brilliant. Soft storage with parenchyma, flexible cartilage with collenchyma, and dead, rigid armor with sclerenchyma. Now that we understand the packing and the supports, we need to look at the actual plumbing system of the plant. How do the water and food physically move? The author details this in the next section, introducing the complex permanent tissues. Let's read it.
Read away.
Quote, conducting tissues, complex permanent tissues. How does water reach the leaves of tall trees? How does food prepared in leaves reach other parts of the plant? Plants have specialized conducting tissues called xylem and phloem, together known as complex permanent tissues, because they are made up of different types of cells working together.
This is a masterclass in tissue complexity. The text notes these are complex permanent tissues because they are made up of different types of cells working together. It is not just a uniform block of simple parenchyma, it is a multi-component machine.
And the two major pipelines are xylem and phloem. Let's break down the water pipe first. The xylem. Quote, xylem is the tissue that transports water and minerals from the roots to other parts of the plant. It also provides strength to the plant. Xylem consists of tracheids, vessels, xylem parenchyma, and xylem fibers. Tracheids and vessels are tubular and thick-walled. Xylem parenchyma are the only living component of xylem, while tracheids, vessels, and xylem fibers are primarily sclerenchymatous.
Okay, its function is to transport water and minerals from the roots upwards. But the architectural revelation here ties directly back to what we just learned about dead cells. The text says the main tubular pipes of the xylem, the tracheids and vessels, are primarily sclerenchymatous.
Meaning they are dead.
Exactly. And logically, this is the only way a highly efficient microscopic water pipe could work. If the cells forming the pipe were alive, they would be full of dense cytoplasm, a bulky nucleus, and a giant vacuole. Trying to suck water through a pipe stuffed full of cellular organs would be nearly impossible.
So the cells form a column, reinforce their walls with lignins so they don't collapse under the immense vacuum pressure of the transpiration pull, and then they die.
The cytoplasm degrades, the ends of the cells dissolve, and they leave behind a perfectly smooth, hollow, dead, continuous tube stretching from the deep roots to the high canopy.
The only living part left, as the text notes, is the xylem parenchyma, which likely sits on the sidelines storing nutrients for the surrounding tissue. So xylem is the dead water pipe. But then the text contrasts this sharply with the phloem, the food delivery system. Let me read that.
Let's hear the difference.
Quote, unlike the xylem, phloem is mostly made up of living cells. It consists of sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Some cells are long and tubular, joined end to end by perforated walls. These cells form sieve tubes. Sieve tubes transport food from leaves to other parts of the plant.
It states explicitly, unlike the xylem, phloem is mostly made up of living cells. We must ask why. Why is the water pipe dead, but the food pipe alive?
It comes down to the complexity of the transport, right? Water in a plant only ever moves in one direction, up, pulled by passive evaporation.
But food, the rich sugars created by photosynthesis in the leaves, has to be actively managed. It needs to go up to fuel new apical meristem growth, but it also needs to go all the way down to the roots to be stored for the winter.
Passive dead pipes can't handle complex routing. The plant needs a living, active system. The text explains that the main channel of the phloem is the sieve tube. These are living, tubular cells joined end to end.
But instead of the ends dissolving completely like the dead xylem, they form perforated walls, like a microscopic colander or a kitchen sieve, allowing the thick, sugary sap to flow from one cell to the next.
But there is a massive biological catch. Let me read the rest. Quote, the cellular functions of the sieve tube cells are regulated by companion cells. Companion cells are specialized parenchyma cells. Main function of companion cells is to monitor loading and unloading of sugars in sieve tubes. Phloem parenchyma store food materials, and resin, tannins, and latex. The sieve tubes are also supported by phloem fibers, which are primarily sclerenchymatous and provide strength.
To function efficiently as a pipe, the sieve tube cell has to empty out most of its interior. It pushes its cytoplasm to the very edges and actually loses its own nucleus.
It willingly gives up its brain.
Exactly. A living cell without a nucleus cannot maintain itself for very long, and it certainly can't manage the complex logistics of routing sugar, which is why the text introduces a vital partner, the companion cells.
The text describes companion cells as specialized cells, whose main function is to monitor loading and unloading of sugars in sieve tubes. The sieve tube is the hollowed-out highway, and the companion cell is the intelligent control tower sitting right next to it.
The companion cell keeps its nucleus. It generates the energy. It literally reaches microscopic chemical arms into the sieve tube and actively pumps sugar in or pulls sugar out, depending on what the plant needs at that exact moment.
It is an exquisite interdependent relationship. The sieve tube provides the transport volume, and the companion cell provides the brains and the brawn to move the cargo.
It is breathtaking coordination. To ensure we grasp how all these simple and complex tissues fit together, the author provides a high-level summary, grouping them into tissue systems, followed by a quiz.
Let's read the exact text for the systems and the questions. Quote, so far, we have studied different types of plant tissues based on their functions, protection, support, and conduction. In a plant body, these tissues do not work alone. They are organized together into larger groups called tissue systems. Plant tissues are organized into three tissue systems. One, dermal tissue system. This forms the outer covering of the plant. It protects the inner parts and reduces water loss. Three, vascular tissue system. This consists of conducting tissues xylem and phloem.
The author is zooming out, showing us the macroarchitecture. The dermal system is the skin, the ground system, which the ellipses replace but implies the parenchyma and sclerenchyma, is the filler and support, and the vascular system is the plumbing.
And then it gives us pause and ponder questions. Quote, 2. Why do you think that a thick cuticle on the outer wall of epidermis is advantageous for a plant living in the desert, but disadvantageous for a plant living underwater? 3. Once water is absorbed by plant roots, it has to travel against gravity through xylem. How do the dead cells of the xylem work together with the living cells of leaves at the top to keep the water moving? 4. What do you think will happen if there were no stomata in the epidermis of the stem or leaves?
Let's test our understanding with those pause and ponder questions. They require us to synthesize everything we just learned. Let's take question 2. Why is a thick, waxy cuticle an advantage in the desert, but a disadvantage underwater?
Well, the desert is an environment defined by the lethal threat of dehydration. A thick, waxy cuticle is the ultimate defense mechanism to lock moisture inside the plant. But if you put that same waxy raincoat on an aquatic plant, you create a disaster.
Right. An aquatic plant is completely submerged. It doesn't fear drying out. In fact, it needs its epidermis to be highly permeable to absorb dissolved carbon dioxide and microscopic nutrients directly from the surrounding water.
A thick wax layer would act as an impermeable barrier. The aquatic plant would essentially isolate itself from its environment and starve to death while surrounded by food.
Question 3. How do the dead cells of the xylem work with the living cells of the leaves to move water against gravity?
This is testing our grasp of the transpiration pull. The dead xylem cells are purely structural. They form the hollow, crush-proof, lignified pipes. They do zero active work.
The work is entirely driven by the living leaf at the top. The living cells open their stomata to the hot sun. The sun evaporates the water. The cohesion of the water molecules creates the vacuum. The living leaf acts as the solar-powered engine, pulling the microscopic rope of water up through the dead, passive structural casing of the xylem.
Yes, a beautiful synergy between dead architecture and living physics. And finally, question 4. What happens if there are no stomata in the epidermis?
Total catastrophic system failure. The stomata are the plant's only mouths. Without them, the plant cannot inhale carbon dioxide, which means photosynthesis stops completely. The plant starves.
Furthermore, without stomata, transpiration cannot happen. The vacuum pull stops. Water stops flowing up from the roots, meaning the plant stops receiving vital minerals and loses its internal hydraulic pressure. It suffocates, starves, and wilts simultaneously.
And with that, we have thoroughly mapped the biological matrix of the plant world. We see how it builds from a dividing meristem down to specialized plumbing. But now, the source text pivots violently.
It turns the microscopic lens away from the leaves and the roots and points it directly at us. We are moving into segment 5, animal tissues. How do the microscopic bricks organize to allow an organism to sprint, to hunt, and to think? Listen to the author's incredibly direct opening to section 3.3.
Go for it.
Quote, 3.3 Animal tissues. Like plants, animal cells also group together and specialized in performing different functions. These groups of similar cells form animal tissues. Let us perform a few simple activities. Blink your eyes quickly. Clench and open your fist. Take a deep breath. Touch something warm or cold.
I love the pedagogical approach here. The author doesn't start with dry definitions. They start with visceral, physical action, blinking, clenching a fist, breathing.
The text continues, quote, now think, which tissue helps you move? Which tissue enables you to sense heat or cold? Which tissue allows oxygen to enter the blood? Which tissue holds the body together so that the skin does not fall off?
When you clench your fist, it feels like a single, unified action. But biologically, it is a symphony of specialized tissues working in perfect concert. Your brain sends an electrical impulse down a specialized wire nervous tissue. That wire interfaces with a fiber designed purely to contract and shorten muscle tissue.
That muscle pulls on a fibrous cable connective tissue tendon, which is anchored to a rigid lever made of calcium connective tissue bone.
The text demands that we stop taking these actions for granted and look at the diversity of animal tissues that make them possible.
Quote, many such questions can be asked. But the answers lie in the diversity of animal tissues, which are specially adapted to perform different functions. It is interesting to understand how the structure of an animal tissue suits its specific function. Let us explore different kinds of animal tissues. Each tissue performs a specific function.
And the guiding principle the author establishes is identical to what we saw in plants. Form follows function. The physical shape of the animal cell is meticulously sculpted to suit its specific job.
The text begins this exploration by examining the tissues that form our borders, the barriers between our internal machinery and the outside world. I'm going to read section 3.3.1. The text presents this information in a detailed table outlining different types of epithelial tissues. I'm going to read each row of this table exactly as written so we can analyze the specific architecture of each lining.
Sounds good.
Quote, 3.3.1 Epithelial Tissues, Structure and Functions. Epithelial tissue forms the outer covering of the body, skin, and also lines the internal organs, such as the mouth, lungs, blood vessels, and intestine. It is composed of closely packed cells with very little space between them. This structure prevents the entry of the germs, reduces.
Let's unpack this baseline definition first. Epithelial tissue is the great liner. It covers the outside of the body as skin, but it also shrink wraps all of our internal organs and lines the inside of our blood vessels. Because its primary job is a barrier, the text notes the cells are closely packed with very little space between them.
They are the tiles on the floor and walls of the body. But the first row of the table describes a very specific type of tile. Let me read it. Quote, single layer of thin, flat cells, lining of the tissue in the blood vessels and lungs. Protection. Protects underlying tissues from mechanical injury, friction, and entry of microbes.
A single layer of thin, flat cells. In anatomical terms, we call this a squamous epithelium. The text locates these thin, flat tiles in the blood vessels and lungs. Why? Think about the function of a lung. Its entire purpose is to allow oxygen in the air to cross over into your bloodstream.
If the wall of your lung was made of thick, heavy, multi-layered bricks, the oxygen molecules would physically struggle to pass through. You would suffocate.
So the body uses the absolute thinnest, flattest cellular tile possible, a single layer of microscopic pavement allowing for instantaneous gas exchange. But let's look at the next row of the table where the architecture completely changes.
Quote, many layers of cells. The outer cells are flat and tightly packed. Skin, mouth, and esophagus. Secretion. Production and secretion of mucus, enzymes, hormones, sweat, saliva.
The text calls for many layers of cells. This is a stratified epithelium. Stratified just means stacked in layers, and the locations make perfect sense. The skin, the mouth, the esophagus.
These are the high-friction environments. When you swallow a piece of hard toast, it physically scrapes down your esophagus. If your esophagus was lined with the delicate single-layer squamous cells of your lungs, the toast would shred the lining and you would bleed internally.
So the body stacks dozens of layers of tiles on top of each other. As the tough, dead outer layers get scraped away by the environment, there are plenty of replacement layers underneath.
It is an architecture built purely for durability and mechanical wear. But then the text introduces a third shape. Let me read the next row. Quote, cells specialized for producing and releasing substances may be cuboidal or columnar, salivary glands, sweat glands, and stomach lining. Sensory functions, smell, taste, sound, and balance.
The geometry changes from flat tiles to three-dimensional blocks, cuboidal or columnar. They look like microscopic sugar cubes or tall architectural pillars.
Why does a cell in a sweat gland or the stomach lining need to be a large cube or a tall column?
Because of the interior volume required to do a job,
Yeah.
the text notes these are specialized for producing and releasing substances. Your stomach lining is constantly manufacturing and pumping out highly corrosive hydrochloric acid. To manufacture complex chemicals, a cell needs a massive amount of internal machinery, endoplasmic reticulum, Golgi bodies, mitochondria.
A flat, pancake-like squamous cell simply doesn't have the physical interior space to hold an acid manufacturing plant. You need a big, chunky cube or a deep column to house the factories.
Yes, and the final row of the table introduces one of the most fascinating epithelial modifications.
Quote, specialized receptor cells having hair like cilia, nostrils, taste buds, and inner ear. Absorption, efficient uptake of nutrients, water, etc. Single layer of tall, pillar-like cells, often with hair-like structure, lining of small intestine.
Ciliated epithelium. The text describes hair-like cilia on the surface of these pillar-like cells.
These aren't hairs like on your head. They are microscopic, whip-like protein structures extending from the top of the cell membrane. In your inner ear, these tiny hairs wave and bend in response to physical sound vibrations, triggering the electrical signals that allow you to hear my voice right now.
And in other parts of the body, like the windpipe, these millions of microscopic hairs beat together in coordinated ways, acting like millions of tiny brooms sweeping dust and trapped mucus up and out of your lungs.
The form perfectly follows the function, from a flat tile for diffusion, to a deep column for manufacturing, to a sweeping hair for movement. The source text takes a brief pause here to include two short, distinct asides. The first is a historical note, and the second is a botanical connection. Let me read both exactly as written. Quote, "Meet the scientists. Sipra Guha Mukherjee, in collaboration with S.C. Maheshwari, made a breakthrough discovery in the area of plant tissue culture. Their research led to the development of a complete plant through anthroculture, using an artificial nutrient medium under controlled laboratory conditions. This pioneering work greatly contributed to crop improvement and brought significant progress in modern agriculture."
These notes ground our microscopic exploration in massive real-world impacts. Sipra Guha Mukherjee's work is a direct fulfillment of the goal stated in paragraph 1 of our document, using tissue knowledge for human welfare. By figuring out how to artificially nurture a specialized tissue into a whole new plant, she essentially unlocked the cheat code for modern agricultural yields.
We learned how to manipulate the blueprint. And then there's the ready to go beyond section. Quote, "Ready to go beyond. In young plants, the outer protective layer is a single-layered epidermis. As plants grow older, some cells below the epidermis of the stem develop the ability to divide, act as lateral merosomatic cells, and form the cork cambium. The division of cork cambium cells gives rise to cork cells. Cork cells are dead, compactly arranged, and contain a substance which makes them impermeable to water and gases. This forms the bark of the tree."
It acts as a perfect capstone to our plant biology, explaining a phenomenon we all recognize but rarely understand. Tree bark. We talked earlier about the delicate, single-layered epidermis covering a young green stem. But as a tree's lateral meristome adds girth year after year, that delicate single layer of skin would stretch and tear.
So the plant activates a secondary defense system, the cork cambium. It starts producing cork cells, which the text notes are dead, compactly arranged, and contain a substance which makes them impermeable to water and gases. The tree essentially wraps its expanding waistline in a thick, dead, waterproof, fire-resistant armor. That is what bark is.
Let's bring our focus back to the human body. We've covered the epithelial tissues that line our surfaces. But what holds all those surfaces, tubes, and organs together? How does the body maintain its physical shape?
The text addresses this fundamental question in section 3.3.2. And in doing so, it introduces a category that creates a massive conceptual paradox. Listen to the exact text.
Okay.
Quote, 3.3.2. How are various parts connected in our body? You have read that blood connects different parts of the body by transporting nutrients, gases, hormones, etc. In the same way, bones connect and support the body from head to toe. A tissue that connects and supports other tissues is called a connective tissue. Both blood and bones are connective tissues.
Both blood and bones.
Okay, I have to stop and push back on the core assertion of this paragraph. The text states unequivocally, both blood and bones are connective tissues. How can biology possibly place a red, splattering liquid and a solid, unbreakable white lever in the exact same tissue category? They couldn't be more physically opposed.
It is the ultimate paradox of anatomy until you understand the deep structural definition of a connective tissue. The text explains it.
Let me finish the quote. Quote, "Though both are connective tissues, they differ in composition and consistency. Blood is fluid, while bone is hard. This difference is due to the matrix, which is watery, soft, and jelly-like in blood, but hard, solid, and rigid in bones."
Unlike epithelial tissue, where the cells are packed tightly shoulder to shoulder like bricks in a wall, connective tissue is defined by distance.
Distance.
Yes. In a connective tissue, the cells are widely spaced apart. They're not the main physical component of the tissue. Instead, these scattered cells spend their time secreting a massive amount of extracellular material around themselves. They build a surrounding environment. And the scientific word the text uses for that surrounding environment is the matrix.
Ah, this difference is due to the matrix? So the tissue isn't defined by the cells themselves, it's defined by the stuff the cells float in.
Exactly. And the text provides the perfect comparison. Look at blood. You have red blood cells and white blood cells widely scattered. What are they floating in? The text says the matrix is watery, soft, and jelly-like. We call that plasma. Because the matrix is liquid, the entire tissue is a liquid. It connects the body by flowing through the vascular plumbing, delivering oxygen from the lungs to the big toe.
But then look at bone. A bone cell, an osteocyte, is also a cell sitting by itself, but instead of floating in liquid, it secretes a matrix made of dense calcium and phosphorus crystals. It literally entombs itself in concrete. The test describes this matrix as hard, solid, and rigid. Because the matrix is solid rock, the tissue is solid rock. It connects the body by acting as a mechanical scaffolding.
Two wildly different physical forms, a river and a rock, but identical underlying architecture. Scattered cells suspended in a matrix.
The text highlights this with an activity. Quote, functions of other connective tissues as experienced through different actions. Activity 3.3, let us perform. 1. Perform the actions given in Table 3.4. 2. Record your experiences and compare them with the experiences given in Table 3.4. 3. Study their functions and identify the connective tissues. Table 3.4, connective tissues. Action, touch your elbow gently. Experience, a hard and rigid structure. Function, give strength, support, and protection. Identified connective tissue, bone.
When you touch your elbow, you aren't just feeling a solid piece of dead plastic. You are feeling a living connective tissue maintained by millions of hidden cells orchestrating that hard, rigid matrix to give you strength and protection.
And recognizing that these hard matrices are alive is crucial for understanding how to maintain them. The source text concludes its exploration of animal tissues by directly linking this biological reality to human lifestyle. Let's read this final bridge. Quote, bridging science and society. Yoga, described in ancient Indian texts, includes physical postures, breathing, and meditation. Research shows it improves flexibility, posture and breathing, reduces stress, and helps prevent lifestyle diseases. Every year, 21st June is observed as the International Yoga Day to promote role of yoga in health and well-being. Maintaining correct posture, proper nutrition, regular exercise in yoga keeps our bones strong, muscles fit, joints flexible, and protects the body from stiffness.
It might seem like a sudden jump to go from discussing the calcified matrix of a bone cell to discussing an ancient physical practice like yoga, but mechanistically they are perfectly linked.
Absolutely. The connective tissues we just discussed, bone, tendons, cartilage, are not static. They are highly responsive living ecosystems.
If you sit in a chair for 10 hours a day, the cells inside your connective tissues sense the lack of mechanical stress. They interpret that lack of movement as a lack of demand, and they will slowly stop reinforcing the matrix. The matrix becomes brittle, the joints stiffen, and the lifestyle diseases the text mentions begin to set in.
But when you engage in the physical postures of yoga or any regular exercise, you are applying intense, dynamic, mechanical loads to your tissues. You are literally pulling on the microscopic matrix. The cells feel that tension, wake up, and actively secrete more collagen, more calcium, and more lubricating fluids. You are biologically commanding your tissues to maintain flexibility and strength.
We have mapped the complex rules of differentiation. We've seen how cells graduate to become permanent fixtures, a rigid piece of bone, a flat lung tile, or a hollow water pipe. But the text saves its most mind-bending revelation for the very end.
We are entering segment 6, the ultimate superpower. What happens when the ironclad laws of cellular graduation are completely broken? Let's read the section titled, Think as a Scientist, exactly as written.
Let's do it.
Quote, think as a scientist. From one cell to an organism. Totipotency. In 1958, F.C. Stewart demonstrated that even single cells from vascular phloem of carrot retain the ability to regenerate whole plants. He was the first person to do so.
This is not just a neat biological trick. It is a fundamental disruption of how we view the finality of life's building blocks.
Let me make sure I am grasping the sheer scale of what F.C. Stewart did in 1958. We established that the phloem is the permanent, complex tissue responsible for moving food up and down the plant. A cell in the phloem is a mature, specialized adult. It has graduated. It is locked into its career as a food pipe.
Correct. Under normal circumstances, that cell will die as a phloem cell. It will never divide again.
But let me read the rest of the experiment. Quote, he and his team performed an experiment using the phloem and cells of carrot. He grew these cells in a nutrient medium containing simple sugars and hormones under appropriate conditions. He observed that these phloem cells divided to form a mass of cells, which gradually divided and differentiated into a complete plant.
So the text says Stewart isolated a single specialized phloem cell from a carrot, put it in a glass tube with some sugar and hormones, and that single adult cell grew into an entire brand new carrot plant. Roots, shoots, leaves, everything.
He essentially triggered a biological time machine.
And the text provides the exact vocabulary to explain this miracle. Let's hear it.
Quote, it shows that cells of phloem first dedifferentiated. cells regain the ability to divide, to form an undifferentiated mass of unspecialized cells. These, when grown in appropriate conditions, supplemented with nutrients and growth chemicals, further divided and redifferentiated to form roots, shoot, and eventually the complete plant. Thus, some mature plant cells have the ability to undifferentiate, divide, and redifferentiate to develop into a new plant under specific conditions. This is known as totipotency, and such cells are totipotent cells. This is similar to the ability of a zygote to divide and differentiate into an entire organism.
De-differentiated means going backward.
Yes. In animal biology, when a cell differentiates into a skin cell or a neuron, it effectively locks away or permanently turns off the other 99% of its DNA blueprint. It throws away the instructions for how to be anything else.
But Stewart proved that plant cells don't destroy the blueprint. They keep the entire manual tucked away. When exposed to the right chemical shock, the hormones in Stewart's test tube, that mature phloem cell de-differentiated, it forgot its identity. It abandoned its specialization and reverted back to a blank slate, a mass of rapidly dividing unspecialized cells.
It reverted back into a meristematic state.
Exactly. And once it was a blank, rapidly dividing mass, the text notes it then redifferentiated. The cell started reading the full blueprint from the beginning, assigning some cells to become new roots, others to become new leaves, rebuilding the entire architecture from scratch.
And the text gives us the ultimate scientific term for this capability: totipotency, the total potential of a single cell to build an entire organism possessing the exact same raw power as the original fertilized zygote that began the life cycle. It proves that every single cell in that carrot is holding the complete universe of the carrot inside it, just waiting for the right signal.
It is a breathtaking reality. To ensure we have locked in all these massive concepts, the text provides a rapid-fire summary and a final quiz.
Let's run through the at-a-glance summary, bullet points exactly as written, and we will decode each one dynamically based on our deep dive.
Fire away.
Quote, tissues are groups of similar cells that work together to perform specific functions.
Form follows function, the specialized laborers of the biological city.
Quote, different tissues coordinate with one another to perform life processes in plants and animals.
The ultimate teamwork. Dead xylem pipes pulling water alongside living phloem tubes, pumping sugar.
Quote, plant tissues are broadly classified into meristematic and permanent tissues, depending on their ability to divide.
The rapid-fire construction workers versus the graduated adult professionals.
Quote, functionally, plant tissues can be categorized as protecting tissue, supporting tissue, and conducting tissue.
The waxy skin, the woody armor, and the complex plumbing.
Quote, permanent tissues may be simple, made up of one type of cell, or complex, made up of more than one type of cell.
Solo operators like parenchyma storage versus the intricate interplay of a sieve tube and its companion cell.
Quote, simple permanent tissues include parenchyma, collenchyma, and sclerenchyma.
Soft bubble wrap, flexible pectin rubber, and dead lignin concrete.
Quote, complex permanent tissues include xylem and phloem that transport water and food respectively to all parts of the plant.
The one-way passive water elevator and the two-way active sugar highway.
Quote, animal tissues are mainly of four types: epithelial, connective, muscular, and nervous tissues.
The four major architectural disciplines of the human body.
Quote, epithelial tissue forms the outer covering of the body or protective lining to internal organs to provide protection and other functions.
The flat squamous tiles of the lung and the tall columnar factories of the stomach.
Quote, connective tissue connects and supports various organs and tissues of the body.
The tissue defined by its environment, cells floating in a watery plasma matrix or entombed in a solid bone matrix.
Quote, muscular tissue produces voluntary and involuntary movements for locomotion and other movements in the body. Quote, nervous tissue consists of neurons to receive and transmit impulses and helps regulate body activities. Quote, skeletal system protects organs and provides support. Quote, in our body, movement occurs by the coordination of muscles and bones, musculoskeletal system, under the control of the nervous system.
The symphony required to simply clench a fist.
Now, the ultimate test. Let's tackle the first three questions from the Revise, Reflect, Refine quiz section. I will read question 1 exactly as written. Quote, 1. Meristematic tissues divide repeatedly. What property of their cells allows them to do this? 1. They have thick walls for protection. 2. They contain large vacuoles that store nutrients. 3. They have thin walls, dense cytoplasm, and large prominent nucleus. 4. They are functionally differentiated cells.
We can deduce this purely through the logic of minimalist engineering. A cell designed for speed cannot afford heavy armor or massive luggage.
Right. A thick wall would take too much energy to split. A large vacuole is a giant waterbed dragging them down, and they certainly haven't differentiated yet, so the answer must be three. They have thin walls for easy splitting, dense cytoplasm for energy, and a large prominent nucleus to rapidly copy the blueprint.
Spot on. Let's hear question 2 exactly.
Quote, 2. If a plant is unable to transport food from leaves to roots, which tissue is malfunctioning? 1. Xylem. 2. Phloem. 3. Epidermis. 4. Sclerenchyma.
The key operational word is food. The plant is failing to move the sugary products of photosynthesis downward.
We know xylem is the dead plumbing for water moving exclusively up. Epidermis is the skin. Sclerenchyma is the dead structural armor. The living food highway, monitored by the companion cells, is the phloem. The answer is 2.
Excellent. The final question exactly.
3. Why are the epithelial tissues that line an animal's internal organs usually only one or a few cells thick? 1. To store food efficiently. To provide maximum strength. 3. To allow quick exchange of materials across them. 4. To reduce friction.
Think back to the squamous tiles lining the microscopic air sacs of the lungs. The body intentionally sacrifices maximum strength in those locations.
Because if the wall is thick, the oxygen cannot physically diffuse through the barrier fast enough to keep the organism alive. The thinness is a feature, not a bug. The answer is 3. To allow quick exchange of materials across them.
We have thoroughly decoded the text, but the author leaves us with one final practical prompt in a section called The Journey Beyond.
I'll read it exactly. Quote, "The journey beyond. Visit a doctor and find out what happens in ligament rupture, cartilage rupture, and fracture of bones. How can we reduce the risk by changing our lifestyle and nutritional balance?"
The text ends not with a microscopic fact, but with a call to action. We now understand that a ligament or a bone is a connective tissue matrix maintained by living cells. When that matrix ruptures, the cells must work overtime to secrete new matrix to bridge the gap. By managing our nutrition and mechanical loading, we dictate the strength of that matrix before the rupture ever happens.
We have officially reached the end of our source document. We have traversed the microscopic landscape from the relentless division of the apical meristem pushing roots through the dark earth to the mind-bending physics of transpiration pulling water hundreds of feet into the sky. We've examined the architectural perfection of a flat epithelial lung tile and the concrete matrix of a human bone.
And as we close this deep dive, I want you to carry forward the staggering implications of that single carrot cell FC steward placed in a test tube. Tutuptancy. The reality that a mature, specialized cell possesses the hidden capacity to hit rewind, forget its highly specific job, and regenerate an entire organism from scratch. Human cells generally lose access to this superpower the moment they differentiate into a liver cell or a bicep. They lock the door to the master blueprint.
But the biological machinery for a cellular time machine exists. The software is written in nature. What happens if 50 years from now, human medicine figures out the exact chemical whisper required to unlock that door in us? Could we take a single scraped-off skin cell, de-differentiate it, and command it to regrow a severed spinal cord or a failing heart? When you understand the sheer potential locked inside the microscopic bricks that build you, the future of human biology doesn't just look promising, it looks limitless. Thank you for joining us on this deep dive into the architecture of life. Keep questioning the incredible engineering inside you.